Hello everyone, my name is Surendra Singh. I am Assistant Professor and Station Director at WSU's Dryland Research Station. I joined here in 2023 and a little bit about the Lind Station. It has got quite a bit of legacy. It's now over 110-year-old and continuously providing growers with research-based information to make a decision on their own operations. And a little bit about the area where Lind Station is located at. It's located in Adams County in eastern Washington, also known as Dryland region of the Pacific Northwest, and it falls under low rainfall precipitation zone, where average rainfall is about 9.5 inches a year. And due to this low moisture condition, the common cropping system is wheat fallow rotation, where wheat is grown in year one, where land is left fallow in year two, and it's back into wheat in year three. So basically, growing one crop in 2 years. Because of the low moisture condition, and it allows soil profile to recharge with the with the rainfall water. And also, while the wheat fallow rotation provide quite reliable wheat yields over the past 100 years, it has also presented some challenges such as declining soil organic matter, herbicide resistant herbicide resistant weeds, heavy soil erosion, and ultimately declining the overall soil health. And since the moisture is really low, there are not a whole lot of options we have. Basically, most to all growers are tied up into three options: either reduce the tillage, reduce the fallow period, or add organic matter directly. And we'll touch base on all of three options and how our trials showed the soil health responses to that. And a lot of the farmers have switched into reduced tillage or no tillage system altogether. And also a lot of them are looking into options to add a little bit more organic matter directly application. And while that provide a little bit of solution, it's also economically challenging to do both, as equipment is expensive and adding organic matter directly to the land is also expensive. And And when I joined in 2023, and this is the first case study where organic matter was added directly, and how soil health looked under that. So when I joined in 2023, I saw these a few lush green bushes of wheat as compared to the surrounding wheat around. And when we dig it deeper, we found out that this was an old biosolid trial which existed from 2011 to 2011 to 2019, which received biosolid application in 2012 and 2016. And that biosolid rate was applied at 2.8 dry ton per acre. And also had a tillage treatment incorporated into it. One is disc tillage, which was referred as intensive tillage, and undercut tillage, which kind of sweeps under the surface and still leaves a significant amount of crop residue on top. And biosolids were compared with synthetic nitrogen and synthetic fertilizer, which is the common practice growers in the region use. And we went ahead and the sample the topsoil and try to find the reasons why it is showing that lush growth. And we found out that the macro micronutrients from the old biosolids, and this sampling was done in and 2013. So that will be 7 years after the last application of biosolids or the last application of those tillage treatment was done. And we saw that, as seen in the table, that soil organic carbon, soil organic matter, organic nitrogen, everything was still significantly higher from the legacy biosolid trials, biosolids treatments, and same goes with the macro micronutrients are still higher, and some of them were actually decreased, which is magnesium and pH, which showed decrease with the legacy biosolids effect. So summarize to say is like there are still all the effects are present which were expected to be happening just during the active phase. And that kind of tells us a little bit about the legacy that even the older management uh, leads to significant differences in soil health. And when we saw the tillage treatment, which was still showing even 7 years after the last treatment application, and organic matter was significantly higher, and magnesium was significantly higher under the undercut. So And we also saw that in the yield, which was taken in 2023, 7 years after, and in 2025, 9 years after the last application. And while tillage didn't affect the wheat yields directly, biosolids had pretty prominent effect. And on an average, in both years, there was on an average about 500 pounds an acre increase from historic synthetic fertilizer application. And and in the ninth year, which was last year, when we took the soil samples again, we saw that soil organic carbon, nitrogen, phosphorus, and sulfur all were also significantly higher. And this is the ninth year after the last application. And this also reflected into the soil microbial community structure, which is also very important component of soil health indicators. And we saw that total microbial biomass, total bacteria, total gram positive, total gram negative, total fungi, and arbuscular mycorrhizal fungi. All of them were significantly higher under the legacy biosolids application. And that prompted us, and when we presented these results to our growers, they were also interested that and it was quite exciting, and they were also interested that how the biosolids compare with other organic amendments such as composted biochar, and rock phosphate, and biosolids at a little lower rate compared to the last one. So we started this trial last year, and and applied all those treatment. And we are looking to collect crop data, soil health data, soil microbial community structure, different carbon fractions, active and as well as intermediate pool as permanganate oxidizable carbon, soil moisture profiles, and this is the trial which led from the legacy effects of it. And moving on to the second point I was mentioning as a potential solution to the soil health in the region, which was diversifying the crop rotation, reducing the fallow period, and reducing the tillage. And this is one of the long-term trials started by my predecessor, Dr. Bill Schillinger, in 1997, and also a lot of treatments were added in 2010. And here we can see that rotation one, which is winter triticale, spring wheat, no till fallow, then continuous spring wheat, which is another rotation, winter wheat till fallow, and winter wheat, spring wheat till fallow. And most of these rotation, while they are reducing the fallow as compared to the traditional wheat 2 year wheat fallow rotation, they also are being used to get better weed control in the region as well. And all these crop rotation at different level of biomass per year on an average, so that data is also added there. And while continuous spring wheat produces the lowest, it is also shown in the previous research that it's not economically feasible for this reason, for this region. So not a whole lot of continuous spring wheat rotation in the region. And when we look at different soil health indicators, uh, we saw that organic matter, respiration, epoxy, extractable organic carbon, total water extractable nitrogen, aggregate stability, and potentially mineralizable nitrogen, all of them were significantly responsive to these improved biomass input and reducing the fallow period compared to the till fallow systems. But other indicators such as beta glucosidase, total nitrogen, beta glucosaminidase, extractable organic nitrogen, 4-day respiration, that did not show much difference among the treatments. And a few of those indicators we'll discuss here in detail. As I mentioned, that I've added the biomass data just to show that how well it correlates with that, and that is the limiting factor, tying back to the initial discussion that on a low moisture condition, there is not enough biomass going back into the soil, which can improve the soil organic matter. And here we also saw that rotation 4, which is 3 year rotation, which has fallow every 3 years rather than every other year, as compared to which is common wheat fallow system, and that is adding the highest biomass levels uh, as compared to the other treatments. And you can see that spring wheat, continuous spring wheat, while not being economical, it still have living root present, and there is no fallow. So there is quite a bit of build of organic matter there. but since it's not economic for the region, there is not much adoption of that. And farmers are using the 3-year rotation quite often to get better handle on their weeds. And a similar trend we saw on permanganate oxidizable carbon as well, that the rotation 4, which is 3-year rotation, had much higher epoxy levels than the control, which is winter wheat till fallow. And one another thing to notice here, that biomass addition per year add more to the correlates more to the soil health indicators compared to the tillage, and which highlights the fact that biomass might be the first uh, lower biomass addition might be the first roadblock of soil health improvement, and by reducing a fallow period and adding more biomass, we can achieve that. And there was little different, but overall the similar trend, that the rotation 4 had the highest microbial activity measured by a 1-day respiration uh, indicator, and other three were not different from each other. And another potential solution or potential land use we have in the region is called conservation reserve program. In that program, farmers retire agriculture land into conservation reserve program for over 10 years and get incentivized for that. And while soil recovers, and it's sown into native grass seed mixtures, and it improves soil health over time. So the main question we are trying to address from this one is that what happens when we transition our ag land into CRP, and how those soil health benefits and soil health indicator response and how those build up over time, versus when we take out from long-term CRP back into agriculture. And that is, both these transitions are happening quite commonly in the region. So that's what research question we are focusing here, that we established this trial last fall, and we are looking to see what happens to soil health indicators, microbial diversity, abundance, and also going a little bit deeper into how microbial functions change based on the targeted metagenomics, and find it deeper, in addition to the soil health indicators such as organic matter, different carbon fractions, and also looking into crop data, how different, what are the yields looking like, how is the weed pressure, what is the grain quality. And with that, I would like to thank the organizers for giving me this opportunity to present here, and also focusing a little bit deeper uh, into the soil health indicators in the dryland region. And with that, please join us on our next field day. It's one of the largest for the dryland region. And please use this QR code to provide your feedback on this talk. And please feel free to reach out to me if there are any questions. Thank you.
My name is Tayler Ulbrich, and I'm based at the Kellogg Biological Station in Southwest Michigan. And today I'll be presenting on long-term science and how it informs soil health and on-farm opportunities. I have several co-authors to thank, and also the organizers for inviting me today. So just for a brief overview of the talk, I'll be talking about briefly introducing our long-term experiments and sharing two stories. The first will be about how do soil carbon pools respond to tillage and cover crops after 25 years. And the second story will be about how can soil health be made accessible for farmers. And then I'll share a bit about future directions for us. And this map just orients you on where most of you probably are and where I am here in Southwest Michigan. So we have 3 long-term experiments on agroecosystems here at the Kellogg Biological Station, and I'm just going to feature 2 of them. The LTER, or the Long-Term Ecological Research Program, started 38 years ago, features a corn, soy, wheat rotation. We have a gradient of management intensities focused primarily on tillage, cover crops, reduced inputs, and we also have annual and perennial crops in this managed system. And the newer program, which I'm working for, is the Long-Term Agroecosystem Research Program, which is funded by the USDA. We joined in 2022, and we're part of a national network. And this experiment seeks to compare an aspirational agricultural system, so something that is more regenerative and achieves more environmental, societal, economic outcomes, to prevailing practices or business as usual. You can see briefly what that looks like for us and KBS at the Kellogg Biological Station. And importantly, this experiment and questions are co-produced with stakeholders. So stakeholders and scientists work together from the very beginning to envision what this experiment and what data collected on this experiment should be. And one other important note is that this is one of the first long-term experiments with modern soil health testing from the beginning. And so from the very beginning, we're measuring soil health indicators that are newer to our science. So to jump right into story one, how does soil health respond to tillage and cover crops after 25 years? I'm going to be sharing this story from two different lenses, looking at different pools of carbon. So first, we'll be focusing on the passive pool of carbon, soil organic carbon, which is much slower to accrue, but lasts a lot longer. And labile carbon or mineralizable carbon, which is microbial respiration, plant root respiration. Intermediate carbon, which is short form is POXI, permanganate oxidizable carbon, which is recent studies are showing is kind of more of a precursor, an early indicator of what will then become this more passive pool. So I'll be presenting our results for these three different pools. So first, for soil organic carbon, this study was done by Kiro Cordova, and she compared 25 years of data from the right near the beginning of this experiment to later on in the experiment in this LTAR site. And so what I'm going to be comparing is how no-till, reduced input system with cover crops, organically certified system with cover crops, and I'll note that that doesn't have manure or compost or synthetic chemical inputs, compares to managed perennial systems with poplar, alfalfa, and conifer. And so what we see here, and I'll already point out from the very beginning that continuous tillage isn't even on this figure because it had soil organic carbon loss after 25 years, whereas continuous no-till had gains, but so did systems that were tilled but had cover crops present. And so continuous tillage causes loss. Even with tillage, living roots significantly increase soil organic carbon. And in the paper, they talk about how the accrual was greatest in the first 12 years. And if we look at the perennial systems, we'll see that overall they have even greater gains in soil organic carbon. Now, I wanted to share just a couple other key results that have come out of this study site about tillage and soil health. Now, we found that no-till systems are consistently, there we go, out-yielding tilled systems after 15 years. And that's especially when it comes to droughty years, because those no-till systems can hold on to soil moisture much longer. And secondly, studies have found that even one tillage pass can lose years of soil organic carbon gains in unmanaged conservation lands or CRP landscapes. And so that's just to show that when we think about the comparisons of tillage versus continuous no-till versus intermittent tillage, we're likely going to see very strong differences in soil organic carbon gains. So if we move to those other pools of carbon, intermediate and labile pools of carbon, this work was led by Christine Sprunger and Tabisha Martin, and they did soil tests on archived soils from 1991 compared to 2021. So this is actually a 30-year comparison, and they looked at that intermediate pool of carbon, POXC, and also mineralizable carbon, which is much more labile and is in direct response to microbial activity. And I will just note that this work was done in partnership with the Environmental Defense Fund. So this is just a plot to briefly show you what you're going to be seeing in these figures. And so POXZ on your axis, that's your intermediate carbon, and across the x-axis are the different cropping systems: conventional till, no-till, reduced input, biologically based, so that reduced input tilled with cover crops, poplar, switchgrass, early successional, and mown grassland. And the orange boxes are from 1991 and the blue are from 2021. And so what we're looking for is an increase if we see that POXC has increased or that carbon pool has increased over 30 years. And so I'm going to use little red arrows to indicate that to make these figures easier to look at for you. So what we see here is that conventional tillage, you have significant losses in POXC, and we have gains in this intermediate carbon pool when you have continuous no-till or you have cover crops in a tilled system. But our perennial systems, for the most part, had even a higher baseline at that initial sampling point, which was just several years after establishment, and they had even greater gains in this intermediate pool. And then if we look at the more active pool of carbon, this microbial respiration, mineralizable carbon, we see similar patterns. But in this case, even the continuous no-till had drops in mineralized carbon. And so what this is telling me is it's that living root in this reduced input and this biologically placed with this cover crops. It's that living root that's really causing the buildup of that mineralizable carbon, that microbial activity. And for our perennial systems, we see that all of them gain significantly after 30 years. So for this first story, we show that after 12 years, no-till and cover crop systems accrue more soil organic carbon than tilled systems. The intermediate and labile pools are more sensitive to management than soil organic carbon. And I'll note that that's what most soil health tests or soil tests and farmers get tests for. And so they're getting this test that's not very sensitive, perhaps, to their management, at least in short time spans. And perennial systems have more and faster accruing carbon pools than annual systems. So how can soil health be made accessible for farmers? First, we're going to look at a study that came out of my PhD. It was just published. I encourage you to check it out. And here we use mixed methods surveys, cognitive mapping or mental modeling, and interviews to ask Michigan farmers, how do they understand soil health? How do they manage their soil health? And how do they evaluate soil health? And just for the sake of time today, I'm just going to be presenting on how do farmers evaluate soil health. We found with the survey that 77% of Michigan farmers use tests to measure soil health, at least sometimes. But then in interviews, it was clear that these are primarily soil nutrient tests, perhaps for that soil organic carbon, but only 2 farmers reported using modern soil health tests for biological activity. And while they think these soil health tests were great because it was interesting, they weren't sure what they learned. And so that's just a key thing for us scientists to think about is, we're producing this data, but how is it usable for farmers? And so then we asked, how does soil health differ on your fields and how can you tell? And primarily we're finding that Michigan farmers are using crop characteristics, yield, height, coloration, and soil characteristics such as pH, compaction, visual ponding, and soil type and texture to describe soil health on their fields. And these here are pictures from our LTAR site, only 2 years after our shift to regenerative management. So on the left, you have our conventional business as usual treatment. On the right, you have the aspirational treatment with the regenerative practices, 5 crop rotation, continuous no-till. And we had an intense rainfall in spring of 2024, where 1 inch of rain fell about in an hour. And we saw incredible washouts in our conventional system, but not in the regenerative system. So this is just showing that visual indicators matter. This is what farmers are largely using, and they respond quickly after to management practice. So the other thing that farmers think about is soil type and texture. So with vastly different soil types, what equals a good soil health value for one versus another soil type? One farmer characterized and said this very clearly in an interview. He said the use of the word health kind of implies that some soil is not productive, it's not healthy, and I'm not sure that's it. It's a different kind of soil. It's not intended to be as productive, It's not going to be as productive, That's not to say it's unhealthy. And so this really resonated with me as he was getting at, one farmer even said, my soil in Michigan can't compare to soil health in Iowa. It's just a different thing. And I don't want to feel bad that mine feels unhealthy when I compare it to Iowa. That really resonated with me. And as scientists, we need to consider that. And so this is work that was led by Christine Sprunger here at MSU. She did this work for 195 farmers across the Eastern Corn Belt, took on-farm soil tests. And what these figures are showing is that for 3 different soil textures, coarse, medium, and fine, the mean value for where they rest for organic matter on the left, or POC, that intermediate soil carbon on the right, vary pretty significantly. And so a farmer with a coarse soil type should not be expecting himself to get to the same levels of organic matter as a farmer with silty or fine soils. And we need to have that characterized in soil health database so farmers have a trusted benchmark that they can try to achieve too. So then how can soil health be made more accessible for farmers? We need to create local benchmarks to give farmers realistic goals for their soil health and management. And we need to identify how farmer methods and desired outcomes align with management changes in lab-based indicators. So how are those visual indicators that farmers are using aligning or comparing with the lab-based metrics we're using in our labs? So some future directions, really briefly, we're really thinking next to scale on farm. So we're asking questions like, how can our long-term results be made more regionally relevant? We're one site that has 30-year these 30 years of data, but is this what's found across different soil types, across different cropping systems? What results are robust at what scales? And how can farmers' methods of evaluation inform or complement lab-based metrics, as I was just saying? And here's just a brief figure. This is a study that is funded by the MSU, Michigan Department of Ag and Rural Development Agricultural Farm Resiliency Program, led by Dr. Christine Sprunger. And here they sampled 91 farms for soil health, 182 fields to really start getting this Michigan-specific soil health benchmarking. And then for 2 field seasons, we're working with 25 farmers, really in depth to collect soil health data, greenhouse gas data, yield data, and farmer interviews to see how soil health, yields, greenhouse gases, and farmer well-being interact and what management might be driving the best outcomes in all of those areas. So with that, I'll end and say thank you to the Sprunger Lab who contributed a lot of this data, amazing field crews throughout the years, and thanks to our funders. And here's my email. So with that, I'll take questions. Thank you so much.
Great, thank you both for those presentations and welcome everyone to the discussion portion of the long-term soils research session for SoilCon 2026. My name is Dani Gelardi. I am a senior soil scientist and climate coordinator with the Washington State Department of Agriculture, and it is my pleasure to moderate today's discussion and to introduce today's panelists. Dr. Tayler Ulbrich is the Associate Director for Engagement at the Kellogg Biological Station Long-Term Agroecosystem Research Program, based at Michigan State University. Our second panelist, Dr. Surendra Singh, is an assistant professor at Washington State University, where he serves as the director of the Dryland Research Station and of the Washington Oilseeds Cropping Systems Program. Greetings to both of you. So as everyone just heard in their presentations, our 2 panelists are experts in how agricultural soils change over time and what those changes mean for productivity, resilience, and sustainability. So let's do a deep dive together into each panelist's research and outreach work at their long-term research sites. First question, can you both provide examples of impacts that were not present in the first few years of your study, which required this long-term view? Or conversely, effects that you saw initially, but that disappeared over time? And Tayler, why don't we start with you?
Yeah, sure thing. So one of the results I shared and can talk a little bit more about is it took us 15 years to see that a continuous no-till system was consistently out yielding a conventional tilled system. And so for the first 15 years, those yields were about stable, but it really took that soil to build long-term soil aggregate stability and build water holding capacity. And at that point, those soils had much better resilience to drought and made it so, especially during drought years, but basically every year after that 15-year point, those continuous no-till systems always were out yielding the conventional tilled systems. And, we also look at the economics of those systems and we see that, right around that 15-year mark too, is when a farmer is most likely to have already paid off whatever cost it was for them to purchase the right equipment to do a no-till system. And at that point, their system is also already paid off that no-till and then profiting above what they would if they were always doing a conventional tillage system. And so it really took that long-term view to be able to see that. And we wouldn't have done that if, one, we were doing intermittent tillage, which a lot of farmers, especially around here in Michigan, do, where they maybe till before one crop but not the other. We wouldn't see that because it really takes that continuous no-till, or if we would have stopped that study after 6 years. So that's one result that I'll highlight here.
Yeah, and I think that's really interesting. And when, as mentioned, like I was in Tennessee for my PhD, we also noticed that in the beginning, when moisture is fine, fertility levels are fine, you don't see as much effects of no tillage and cover crops. But when drought happens or moisture or anything goes out of the optimum range, that's when those effects on soil resilience from these practices start to show up. So we did notice that in one of the trials. And here in Dryland, basically, it does take long term in general, even in good rainfall zones. But here we have like less than 10 inches a year. So every other year is a fallow period. So it definitely takes about double than what it should be in higher rainfall zones. So that's for like the bottom line is we do need long term research we won't be able to notice any significant improvement in shorter terms in general. However, having said that, there are some of the indicators like soil microbial indicators, microbial stress indicators, those start to show up, and potentially some of the labile and more sensitive carbon fractions also tend to show up, but that still will take a few years to build up in the soil. So yeah, that's what we noticed here. So yeah, definitely long-term soil health research is potentially the only way to go in that.
Yeah, I'll just add, Surendra, I think that's a really great point. And one thing that, we've seen too is that soil organic carbon, which is what most farmers are probably getting measured on their soil, takes a really long time to respond. Like we didn't even see it start to respond for probably at least 4 to 5 years, and it took 12 years to really start to see a difference in those systems. Whereas, like you said, those labile measurements or that microbial respiration, those more labile carbon pools can respond much more quickly. So farmers getting those tests done might be able to see responses to their soil health is really, in a much shorter time frame, and give you a little bit more satisfaction that you're doing something good for your soil health, whereas soil organic carbon alone won't tell you. So I think that's a great point you brought that up.
So we just talked a little bit about how long it can take for some soil health indicators to change, but I think, Taylor, you really kind of zoned in on an area of great interest with the yield measurements and how long those can take to really start to see results. For a researcher who gets the great fortune to look at a soil for 15 years and sort of wait to see that, that's really amazing. But how do you talk to a grower about it who maybe has to make economic or profit-driven decisions year to year and can't necessarily afford to wait 10 or 15 years to start to see the payoff?
Yeah, that's a really good question. I mean, right? Saying that you got to wait 15 years to see a system out yielding. We've starting to, I've heard a lot of farmers too that make the decision to go to continuous or to go to no-till is that it saves them time and it saves them money up front, especially if they're able to use like a modified tractor of some sort. You don't get the newest, fanciest no-till machine, because you're not spending money on gas. You don't have to do those passes before every single season. And so we've had some farmers say, like, I just don't have to be in the tractor as much. So there are some other social benefits that come with doing no-till that I think help farmers wrap their mind around it, or thinking about other crops in your system that you could do. If you really don't want to invest in a no-till system, we've also seen in our research that cover crops do a world of wonder for soil health, and that those soil organic carbon numbers were increasing even in a tilled system when they had cover crops. And so I think that we don't do it justice if we tell farmers that they just need to no-till all the time, because not every soil type is going to work well for that. There are reasons why a farmer might need to till, but then there are other things you can do like cover crops that maybe helps you get through that hurdle or that time frame of I'm trying to build up my soil health, but I'm not yet at the point where I can do continuous no-till for XYZ reason.
True. And even here, it's a low moisture conditions where I'm located, and in general for the dryland Pacific Northwest, cover crops are a challenge to figure out, because if we are doing like one cash crop in 2 years, so profit margins are super thin to work with. And then we are growing one cash crop in 2 years because of low moisture, and growing cover crops sometimes take up that useful moisture. That's where
Yeah.
the profit lies in. So it's a hard sell, and it's very difficult to figure out agronomically and economically. However, growers have put over the past 100 years a humongous effort into reducing the tillage and saving the soil from heavy wind erosion in the region. However, like not a whole lot of rainfall areas are still compatible with the wiggle room growers have to go complete into no-till. So, growers who could get into the no-till system, they did already, and it is, to say the least, it is an investment. Uh, and Obviously, the machinery wise, it's expensive. Then you have to pretty much reprogram the whole operation according to that, and especially if you're working on 2 year rotation, 3 year rotation, and and then diversifying that with, if there is any other crop like other than wheat, if there is any other crop which has to be included like canola, peas in the system, that's always complicates the operation in longer terms. So that's the conundrum and potentially drawback. And another one, what in the Pacific Northwest we started to notice is that no-till system who are completely no-till rely heavily on chemical control on the weeds. And slowly we have started to see that the major weeds are getting resistant to the major chemistry of the herbicides used. So then there is occasional tillage comes into play. A lot of farmers are doing that, but that's just like the what do you say? We'll have to find that sweet spot where
Mhm.
economically it is good versus it's not developing more herbicide-resistant weeds, as well as it is saving the soils from getting more degraded and eroded in the region. So it there are different Venn diagrams following up. So we kind of have to hit that sweet spot on where we can optimize our operations without being too much without being too much loss on the profitability.
Thank you. So you both clearly have a lot of experience working with farmers as well as working with researchers. And I'd love to ask you each for a piece of advice for both of those groups. So we'll start with the farmers. Can you describe a finding from your research that growers can use in their day-to-day decision making to build soil health?
I guess my response here in Michigan would be to plant cover crops. Our research has really said to plant cover crops, but clearly, we get 40 inches of rain a year. And so that's a lot more than the 10 inches that Surendra's area gets. And so, it's all context specific. But I think another thing that we're starting to see in our research, especially as we go on farm, is that there's a need for these local benchmarks of your soil health to help farmers evaluate what they should be reaching for in terms of these soil health values, especially when we're thinking more about these biological metrics. And so, using that data to inform what you're doing as a farmer, I think can be really valuable. But I'll be honest, I don't think the science is totally there yet either. And so forming these collaborations with scientists and farmers so you can see what is changing over time and how can I keep doing that better. I think there's a lot of growth that can be had through those relationships. So sorry if my cover crop answer isn't very helpful for you in the dry area, but I think for us that would be one clear thing that our research has shown.
Yes, and I was in Tennessee as well. Like we have like about 50 inches rainfall there, so that's like 5 times of what we get get in a year. So obviously that's feasible there. But in general, like here in the region, growers what we found through our research, and that's also in my presentation as well, that diversifying the crop or kind of intensifying a cropping system a little bit can not only help with the soil building and increase the amount of biomass going back into the soil, it also helps take care of weeds a little better than the traditional conventional method, which is wheat fallow, it's going into it. But yeah, I think that's one way, and obviously reducing the tillage not only helps with soil health building, but also saves our soil from long-term erosion as well. So these two main points uh, we pretty much found through all of our research. And in addition, I think as Taylor mentioned, I think it has to be in the collaboration with the growers that what treatments and what is the end product we want, and that we're designing into it. And it's still an evolving area. So I'm really excited for where this collaboration between scientists and farmers lead us.
Thanks. Yeah, I think the point about benchmarks is especially relevant. Taylor, you showed those graphs of texture and how different the metrics are. And this is something I like to talk with growers a lot about. If someone tells you your soil is unhealthy, it's good to investigate that a little bit more. Unhealthy compared to what? What does unhealthy mean? What are the benchmarks that you're describing and how might that be different for me? So it's really nice to see this kind of context-based benchmarking that's happening.
Mhm.
So next, let's talk about a piece of advice for the researchers. Long-term research is the goal of many scientists, but as we've discussed, it can be difficult to fund and difficult to manage. Do you have any tips for researchers and for farm communities on how to secure long-term funding commitments for such projects?
Sure. Yeah, it does take a lot to start a trial and maintain it for a long time, given the funding situation varies from year to year. And most of the funding cycles we pretty much target on the federal state levels are 3 years, 4 year at max, and then we start the trial, but then carrying it over to long term. So my suggestion would be to diversify your funding sources on a basic level, at least just to keep the trials going and continue measuring what the objectives are, and diversifying between federal, regional, commodity boards funding, check off dollars in some states they provide, and also the Department of Ags of the state. So I think diversifying as much as possible on the fundamental level of the trial management and continued data collection. So I think that's really important. I would suggest diversifying, and we did that here as well. And obviously the grower support makes it really worthwhile and easier on us.
Yeah, I think that's a great point. We've been really lucky because the long-term sites here have a consistent funding source, but there's so many side projects that's been essential to our long-term projects here too, to try things out and see what works in these systems. I think kind of two other little nuggets I would mention in terms of helping these long-term projects be successful. One is archive samples, because you might not have the funds right away to process that data. But dried soil, you can still do a lot of analyses on it later. All of those soil carbon metrics that I talked about in the talk were done on archived dried soils. And so there's a lot of opportunity there. If you don't have the upfront costs, save your samples and invest in storage space perhaps, so that you can do data analyses later for those findings. And second, invest in techs that'll stay around. So we have, I would say, a lot of our success of our program has also been because we have several really key people that have been along for the long haul, and they know the interworkings of the systems and the management. And that's hard to do because people move on. But I really do think that's contributed to a lot of our success. I said two, but I think I have one more actually. I think that the way that our systems are moving and our funding sources are moving is they're looking for a clear connection to community need and agricultural community need. And so I'm really excited about that as an engagement specialist, and thinking about, start your project from the beginning with a clear partnership with community members, with farmers that are part of the project and aren't just giving you random input, but are part of the project from the beginning. And I really do think that there's a need and an interest of that from funding agencies. So starting with that model from the beginning might help secure funding sources down the road.
I love that. It sounds like we're talking about diversifying both the funding sources and also the team members, whether that be technicians, researchers, farmers, or others in the community who have a stake in it.
That's a great way to put it.
Yep. And archiving samples. I cannot emphasize enough. Thank you for bringing that up. That is the key. Yes, archiving the samples.
Everyone and their hundreds of jars in their basement of soil. Okay, so next and final question for both of you. It's a big one, so it's okay if you need to take a minute, but what has been the most enjoyable, the least enjoyable, and the most surprising part of your experiment?
Oh, that's a big one. Okay. Yeah, so most enjoyable. I think the most enjoyable is I just love how much learning I had to do to like bring my knowledge up to the level where I can start the trial and on different management practices. And I learned mostly from farmers' interaction. So that has been the most enjoyable, and how passionate people are. When you are in a school, you just think about like that's it. But when you go out in the real world and start doing research on farm, and that's how you learn like why things are the way they are. So the context is really important. So how much I have learned coming from Tennessee, like 50 inches rainfall to less than 10 here. So it was a steep learning curve, but I really enjoyed all of my interactions, feedback, collaboration, the support being offered for the trials, for the station. It's amazing to see. Least enjoyable. Least enjoyable would be that you are going to the field with your equipment and it's breakdown. So and you are working for some deadline data collection, and your lab instruments are not behaving the way they should. So that's being the most I would say least enjoyable part in the journey. And what was the third one? I forgot.
Most surprising.
Oh, most surprising. Hmm. Ah, I think pretty much in research, like when we start a trial or write a proposal, I think most of the time we have a preliminary idea how it might turn out, or just kind of an hypothesis in our mind. But generally, what I found was like it may not turn out that way for the most of the times. So that's being more surprising to me. And one of the results I showed in the slides as well, like the biosolids trial we had, and that was like a surprising discovery as well. Like that during when the trial was going on, we didn't see much of the effect, which is quite surprising because you are actively putting the organic matter in, and that didn't translate into yield for the most years. And now we are seeing consistent increase when the trial was concluded in 2019, revived in 2023, returned back to the normal wheat fallow systems, and now we are seeing year-on-year increase on that. So that was also one of the surprising thing. Like, and not only that, like the tillage part, like we had two types of tillage. One is like disc, and one is undercut, like kind of minimum and intensive. But even after returning that part of the ground to the tilled faction, it is still showing that tillage effect on organic matter that was done in like 9 years ago. So like that is really surprising to me. Like soil remembers whatever you did.
Yeah,
So
I love that.
it's
Yeah, and I really loved the aerial images of where the biosolids had been applied. I can imagine
Mhm.
on the ground you might not see that, but from above it was very, very clear.
It was, and that's how we actually found that trial. That's how we stumbled upon it. It wasn't planned or anything.
that's so cool. My husband does drone flights and aerial imagery, and I studied microbes, and so we're always talking about how we're working at two different scales, but you can really learn a lot at both.
Yeah.
Yeah, so least, most, and surprising. So I'd say most for me is also the people, seeing our project grow and seeing these relationships develop. We have a farmer collaborator now who has what he calls his KBS field because he's like, you guys keep giving me new ideas. I'm just gonna keep trying out these things. So I just that's really enjoyable and refreshing for me. Um, and at the same time, in our LTAR site, we see visual differences in these systems after only 4 years. I showed the pictures of the erosion, and that's also been really exciting because I think sometimes as scientists, we get so stuck on the data, and then it's like, just go out there and look at the
Mm.
soil. And because I'm on, this project just started 3 years ago, 5 years ago now, time eludes me, but it's new and we're already seeing visible difference, even if the data isn't saying it yet in all situations. So that's also been really enjoyable. Least groundhogs and slugs. We have
Oh,
so many of them. We're realizing that because a lot of these fields were conventionally managed and now they're transitioning to this more regenerative system, a lot of, for instance, the predator prey cycles are really out of whack. And so all of the predators that would kill slugs were treated in these, more pesticide heavy systems. And so there's a huge slug population that is decimating our canola and our soy plants. And so that's been really challenging and frankly kind of disgusting. There's some researchers out there that, count how many slugs are like on a shingle to see how many are out there. And I think at one point she said on like a shingle this size, there were like over 100 slugs.
oh,
So least favorite. Um, Surprising, I'd say how hard it is to balance the question earlier, like the long-term need, but also the need to stay updated. A lot of scientists are really excited about the opportunity with our new LTAR system to update it every 5 years. But now that we have 5 years of data, they're like, no, but I'm starting to see a trend and we can't change it because we have to look at how it's gonna go. It's like, yeah, but we also have to update it for the technology. So that's been surprisingly hard to navigate those conversations and figure out how we do that going forward. Um, yeah.
well, it sounds like we're very grateful for the people and maybe less so for the slugs and groundhogs. So, thank you, Taylor and Surendra, so much for this discussion and for sharing about your work. Your perspectives really help highlight the unique value of real-world impacts of long-term soil health research. And it's nice to hear the results from totally different regions, from 10 to 40 inches of rain per year. So with that, we will end today's panel discussion, and I look forward to hearing more questions from the webinar audience.
Thank you
Perfect.
so much.
Yep. Thank you so much.
Hi, I'm Karen Hills. I manage the Sustainable Farms and Fields program with the Washington State Conservation Commission. I will be moderating the live Q&A portion of the remaining sessions. We have a few minutes here for questions and answers from the audience. I know some of you have put some in the Q&A portion of Zoom, and I'm going to see how many of these we can get through. We had question for Surendra. There were a number of people who had questions about testing for PFAS in the areas where biosolids were applied or in the biosolids themselves. So maybe we'll start there.
Yes, of course. And I think this is probably like the fourth biosolid related talk of the month of this year. And we do get a lot of questions about biosolids relating to concerns like PFAS, heavy metals, pharmaceuticals, and how that translate into when you land apply that for food production systems. And just to provide a little bit of background on like what exactly the PFAS are and how they end up in the biosolids, and then what happens after that. So for PFAS, I think it's known as per and polyfluoroalkyl substances. Oh, so and there are about like 150,000 compounds in that category, and out of that we know quite a bit little out of that, like probably like about 70 PFAS compounds can be analyzed with current protocols. So there is a lot to come up from that, and why they end up in the biosolids, because of our daily interaction with the PFAS in terms of food packaging, cosmetics, apparels we wear, firefighting foam, food takeouts, carpets. It's pretty much in most of the daily interactive items, and hence they end up in there. And another thing is like biosolids from industrial waste versus biosolids from residential waste. PFAS vary quite a bit, and residential waste are likely to have way less PFAS concentrations. So that's a little bit of background. And yes, we have started testing on this one. And that is another reason that the new study we started at the station, because if you start testing on the legacy trial, we may not be find the accurate picture of that, because the biosolids last application was 9 years ago. So hence we started the new trial and continue to start testing on the PFAS we can. And yeah, so does that answer the question? And
Yeah, I think that's
yeah.
good. The person who asked can follow up
Sure.
with you directly if they would like more. We're
And
gonna move, actually another one for you, Surendra.
mhm.
There is a question about whether your soil health analyses are looking at soil biology and kind of what aspects of soil biology, if you could speak a little bit more to that.
Yeah, and soil biology is really important, as mentioned in other presentations too, especially from agriculture perspective. But here we are testing for soil biological indicators, and also for soil fungi, bacteria, microbial community structure, and going way deeper into microbial stress indicators. And from the trials, the crop rotation trials and the biosolid trial I presented, we tested on both of them. So we are testing the biology, and so far results pretty much uh, relates back to the how much organic matter and least disturbance we are doing to the soil. So it's pretty correlated, relative of that. And I think that's pretty much it. And this also correlate back to the active fractions of carbon we have tested, the respiration as well. So yeah, it does translate back to that.
Okay, thanks so much. And I'm going to have a question for Tayler here. The question is about the logistics of starting no-till in a corn crop, such as navigating corn stubble. Could you speak to that a little bit?
Yeah, that's a great question. And it's something that comes up a lot. I think in our systems here, it's a challenge, but it's maybe not as much of a challenge as in dry systems because we have more moisture, we have higher decomposition rates, and so it can break down more. I know some of the previous speakers talked about vertical tillage as a way to get into it, or strip till as a way to kind of slowly start decreasing your tillage, but not getting totally to no till. There's also been some research here on short corn and the opportunity that it might provide to basically provide the same yield but have overall less biomass, which then is less residue to break down. So that's some of the research going on. I don't have a farming background personally, so I can't give a lot of detail on the exact practical applications of that, but I do know that there's a lot of research going into trying to figure out how can we make that transition easier, because especially in areas where it's really dry and you don't have that breakdown really quickly, it really can be a challenge to entry. And even here in Michigan, we have a lot of farmers that will do no-till before soy and till again before corn because of that exact same challenge. So it's felt around the country.
Okay, thanks for that. I think we have time for one more question, and this is for Surendra. What specific management decisions are you making based on one-day soil respiration measures? Because of the soil disturbance, artificial moisture, and temperature conditions, can we do better than this method? That's something you can speak to?
Yes, and well, management side, especially from like agronomic management, this directly doesn't affect us a whole lot because our main focus is to stop the soil wind erosion in the area and also increase the soil organic matter input. And respiration is kind of coming out of that. So we necessarily don't decide based on the respiration alone because it has a lot of variability that how much respiration is enough, how much respiration is less. So that's going to be a little bit of too much to take into account while managing field operations. But what we found from different studies across the globe that most of the, if you manage soil organic matter and soil physical properties well, microbes thrive really well because they have enough food to eat and uh, enough organic matter cycling going on, and that also directly relates to the respiration as well. So main focus is to reduce the erosion and increase our organic matter inputs and organic carbon in the soil. uh, yeah, so that's what I would say
Thank
on that
you.
one. Yeah.
Appreciate that. So unfortunately, we are out of time for questions, but I wanted to thank both Tayler and Surendra for their presentations and for the discussion. We will move on to our next session now, but Surendra and Tayler will be available to respond to additional questions in the Zoom chat.